Good evening. Welcome back. This is a space for the part of your mind that won't stop running, the part that keeps cycling through the day, rehearsing tomorrow, calculating what's still unfinished. You don't need to silence it. Tonight, we're going to give it something worth following. Draw a slow breath. Let your body settle into wherever you're resting. We're going to go somewhere very small. Tonight's subject is survival, not the loud, dramatic kind, but the quiet kind. The kind that has been happening beneath your feet for 500 million years, invisible to the naked eye, requiring no drama and no audience. We tend to think of survival as resistance, fighting, fleeing, building walls against what threatens us. But at the microscopic scale, the most resilient life on Earth takes a different approach entirely. It cooperates with destruction. It dissolves into stillness. It waits. The question worth sitting with tonight isn't how these organisms survive. It's why stillness, deep, structural, molecular stillness, turns out to be the most durable strategy in the known universe. Feel your body growing heavier. Your breathing is slowing on its own. You don't have to make that happen. The same automation that runs the chemistry of every cell in your body is quietly doing its work. Our starting point is a cluster of Dicranum broom moss, anchored to the bark of an old oak tree on the western edge of a Norwegian fjord. It's twilight. A cold sea mist is rolling in, condensing into tiny droplets on every green filament. To a person standing at the base of that tree, the moss is unremarkable, a soft green cushion on bark. But shrink your perspective by a factor of 10,000, and the moss becomes something else entirely. At this scale, gravity loses its grip. The dominant forces here are surface tension, capillary pressure, and molecular adhesion, the physics of the very small. Each individual moss leaf rises like a translucent jade plate, and between the leaves, water doesn't simply coat the surface. It's pulled inward by the electrostatic attraction of the plant cells, forming suspended liquid corridors, still clear, perfectly stable channels of water that serve as highways for an entire miniature ecosystem. There are no currents here, no turbulence. The water behaves more like a dense, transparent gel holding everything in place. The geometry of the plant itself creates this stability, each leaf precisely spaced to distribute hydrostatic pressure evenly across the whole structure. It's not a random arrangement. It's the result of hundreds of millions of years of structural refinement. Within these corridors, moving with the unhurried precision of something that has never needed to rush, is a tardigrade. The tardigrade, colloquially called the moss piglet, is less than 0.5 millimeter long. Its body is divided into 4 plump segments, each bearing a pair of stubby legs tipped with 4 to 8 small translucent claws. It doesn't swim. It walks. Each step is a slow, alternating sequence, gripping the moss cell walls with quiet mechanical confidence. Its outer shell, the cuticle, is made of flexible chitin. Under the dim light filtering through the forest canopy, it carries a faint, pearlescent sheen. It feeds by piercing moss cell walls with needle-like stylets, extracting fluid with calm precision. It moves through its liquid environment without urgency, operating on a biological clock that has no interest in acceleration. The moss and the tardigrade are not just neighbors, they are structurally interdependent. The water film coating the leaves is the tardigrade's medium for both movement and gas exchange. Without it, the thin cuticle can't perform respiration. The plant's geometry provides exactly the boundary layer the animal needs to maintain equilibrium. Neither is complete without the other. Each step is a small equation of force and surface, the animal's negligible mass distributed flawlessly across the wet cellulose. There's no heart beating rapidly inside this frame. The slow, rhythmic shifting of its body segments keeps its internal chemistry distributed and balanced. It is, in every sense, a creature that has found total efficiency within a few square millimeters of damp moss. The tardigrade is not alone. Sharing the same liquid highways, navigating the same capillary corridors, are two other organisms from entirely separate branches of the animal kingdom, nematodes and bidoloid rotifers. These three lineages have coexisted in environments like this for hundreds of millions of years, through mass extinctions, ice ages, continental shifts. They don't compete in any obvious way. They occupy the same physical space and face the same threats, each with their own molecular solution. The nematode, the roundworm, is the minimalist of the three. Its body is a smooth, unsegmented cylinder tapering at both ends, roughly the width of a human hair. It has no legs, no appendages, no external complexity. What it does have is a beautifully engineered locomotion system, two sheets of longitudinal muscle running the length of its body, contracting in alternating waves against the pressure of its fluid-filled interior to produce a continuous sinusoidal motion. It moves like a pulse of kinetic energy through the liquid corridors, a silent, hypnotic thread. What makes the nematode remarkable on a deeper level is something called eutely. Every individual of its species has an identical fixed number of cells from birth. The cell count never varies. Its nervous system, one of the most thoroughly mapped structures in all of biology, is a precise, hard-wired network where every neuron has a known function. There is no redundancy, no waste. It is a system stripped down to its essential parameters and then perfected. The bidoloid rotifer is the most visually striking of the three. At its front end sits a double-lobed organ called the corona, fringed with rows of hair-like cilia. When activated, these cilia don't beat randomly. They fire in a metronomic sequence that creates the optical illusion of two spinning wheels. This isn't splashing. It's a precision hydrodynamic pump, pulling suspended organic particles down into the animal's mouth in a smooth, predictable spiral. At its rear, adhesive cement glands anchor it to the moss surface while it feeds. Inside the rotifer, a muscular structure called the mastax grinds incoming particles with hard, chitinous jaws, a rhythmic mechanical thumping that processes food continuously. Every system in this animal operates with the same steady, unhurried beat. These three organisms share the moss as a common space without conflict, each processing their world on a different biochemical frequency, all governed by the same underlying physics. As the night deepens over the fjord, the mist lifts. A dry Arctic wind funnels down from the peaks and begins stripping moisture from the moss leaf by leaf. At the microscopic scale, this is catastrophic. A living cell is not just filled with water. Water is an active structural component. Water molecules form a protective envelope around proteins, maintaining their precise three-dimensional geometry through hydrogen bonding. When water disappears, proteins lose this scaffolding. They begin to unfold, exposing their sticky interiors, and tangle irreversibly. Cell membranes, the oily bilayers that define the cell's boundary, lose their fluidity, stiffen, and crack. The internal machinery collapses. For virtually any organism on Earth, this desiccation is a death sentence. But the three organisms in our moss cluster have had 500 million years to develop a different answer. As the water level in their cells drops below a critical threshold, their genetic machinery triggers a cascade of responses that have been refined across geological time. They don't fight the evaporation. They don't try to retain the water. Instead, they initiate a systematic, orderly shutdown, a biological protocol called anhydrobiosis, life without water. The outer moss leaves sacrifice their moisture first, creating a temporary buffer that slows evaporation in the deeper corridors. This delay matters. It gives the organisms enough time to build their defenses before the last water leaves. The tardigrade's response is architectural. It pulls its head and all 8 legs inside its body wall. It folds its cuticle along specific structural lines, not randomly, but along built-in points of weakness that allow the compression to be even and controlled. The body contracts along its length until it forms a compact, barrel-shaped cylinder, the Tun. This shape change is a precise piece of mathematics. A cylinder has a lower surface area to volume ratio than an elongated body, which dramatically slows the final rate of water loss. Over several hours, the tardigrade sheds up to 97% of its water content, shrinking to a dense, desiccated speck. But the real engineering happens at the molecular level. As dehydration peaks, the tardigrade synthesizes a specialized class of proteins called tardigrade disordered proteins, TDPs. Under normal conditions, these proteins are loose and flexible, moving freely through the cytoplasm. But as their concentration rises past a critical threshold, something remarkable happens. They begin to self-assemble. They interact with one another and spontaneously form a densely cross-linked molecular scaffold that fills every nanometer of the cell's interior. Critically, they don't form crystals. A crystal has a rigid repeating lattice with sharp edges that would tear cell walls apart from the inside. Instead, the TDPs achieve vitrification. They transition from liquid directly into an amorphous solid. The cytoplasm becomes biological glass. This glass wraps every protein, holds every organelle in place, and locks every metabolic enzyme mid-stroke. Nothing can move. Nothing can degrade. Time, in any biological sense, stops. The nematode solves the same problem with a different molecule. As water leaves its cells, they flood with Trehalose, a simple disaccharide sugar with a specific molecular geometry. Trehalose molecules step directly into the spaces vacated by water, forming hydrogen bonds with the surfaces of proteins and cell membranes. They act as structural surrogates for the missing moisture, tricking the cell's internal machinery into behaving as if it's still fully hydrated. As desiccation completes, the trehalose concentration reaches saturation and undergoes the same vitrification transition. Liquid to amorphous solid, cytoplasm to biological glass. Every component is locked in place. Every reaction halts. The cell becomes a transparent, unyielding vault of sugar. Both strategies, protein glass and sugar glass, achieve the same thermodynamic outcome. They prevent the molecular chaos that normally follows dehydration by physically immobilizing every component before disorder can set in. The spatial orientation of every protein, every lipid, every strand of DNA is preserved exactly as it was at the moment of vitrification. Nothing drifts. Nothing tangles. This is cryptobiosis, hidden life. Metabolic activity drops to less than 1/100th of 1% of normal. The organisms are no longer functioning, but they are not dying. They exist as pure structural arrangements of matter, waiting. Inside the vitrified state, these organisms become nearly indestructible. Cool them to -272 degrees Celsius, 1 degree above absolute zero, where atoms barely vibrate, and the glass matrix holds every protein in place without structural damage. Heat them to 150 degrees Celsius, well above the boiling point of water, and the thermal stability of the glass scaffold protects the architecture. Subject them to 6,000 atmospheres of hydrostatic pressure, 6 times the force at the deepest point of the Mariana Trench. And because the interior is uniform, non-porous glass with no water left to compress, the pressure distributes evenly across the entire molecular framework without a fracture. Even ionizing radiation, which would normally destroy DNA by splitting water molecules into reactive free radicals, has limited effect. In the desiccated state, there's no water to split, and if radiation does manage to break a strand of DNA directly, the broken ends can't drift apart. They're locked in the surrounding glass, held in perfect alignment for later repair. The information isn't lost. It's paused. The bdelloid rotifer extends this resilience even further. When desiccation shatters its DNA, the fragments are held in place by the glass matrix. Upon rehydration, the rotifer's enzymatic repair machinery reassembles the broken strands with remarkable precision. But here's what makes the rotifer genuinely unusual. As its membranes rehydrate, they become temporarily permeable, and the animal absorbs loose DNA fragments from the surrounding environment, from the moss, from bacteria, from fungi. It incorporates foreign genetic material into its own genome, using it to patch vulnerabilities and acquire new biochemical tools. The bdelloid rotifer has reproduced exclusively through cloning, no males, no sexual reproduction, for over 40 million years. It shouldn't have survived this long without the genetic diversity that sexual reproduction provides. It has solved this problem by making every rehydration event a genetic renovation, absorbing the adaptive history of its entire ecosystem. It doesn't need a mate. It drinks the evolution of its neighbors. These organisms can remain in the vitrified state indefinitely. In Siberian permafrost, nematodes have been revived after 46,000 years of stasis. In Arctic ice, beetaloid rotifers have returned to active life after 24,000 years. To the vitrified cells of these organisms, that duration is indistinguishable from an afternoon. There was no metabolic activity, no aging, no degradation. The biological glass held everything in place, uncoupled from the flow of time entirely. Permanence, at this scale, is achieved not by resisting the environment, but by becoming indifferent to it. The Tun doesn't expend energy maintaining temperature or pumping out toxins. It allows its internal state to match the ambient conditions of whatever surrounds it, desert, tundra, vacuum, and simply waits. Eventually, rain arrives. A drop strikes the dry moss cluster. The porous plant cells pull the moisture inward via capillary suction, filling the empty corridors with water. The fluid reaches the vitrified tuns and begins to diffuse through the tiny pores of the cuticle. What follows requires no energy from the dormant organisms. It is driven entirely by the physical properties of water, its polarity, its tendency to form hydrogen bonds, its kinetic diffusion. Water molecules insert themselves between the cross-linked chains of the biological glass, gently disrupting the electrostatic bonds that held it together. The glass doesn't shatter, it melts. Smoothly, steadily, the amorphous solid transitions back into a hydrated cytoplasm. Because nothing displaced during the drying phase, because every protein, every organelle, every strand of DNA was held in the same position it occupied at the moment of vitrification, nothing needs to be rebuilt. The cell membranes regain their flexibility. The enzymes, frozen mid-reaction, find their substrates again. The metabolic pathways resume firing. The tardigrade's body wall expands. Its claws begin to flex. 8 stubby legs extend and find the moss surface. The nematode resumes its sinusoidal glide through the restored fluid corridors. The rotifer anchors its foot, extends its corona, and sets its cilia spinning. 2 slow, synchronized wheels generating the same quiet microvortices as before. They return to their quiet work, entirely unaware of how long they were gone. Life, at its most durable, is not a fire that must be constantly fed. It is a structural arrangement of matter that can be paused, and when the right conditions return, it simply resumes. Your body understands this kind of deep pause. Your nervous system is already moving toward it. The analytical loops of the day are losing their urgency. Your breathing is slower than it was when we started. The architecture of rest is assembling itself in the same quiet, automatic way the tardigrade builds its glass. Without effort, without force, without anything required of you. Let the day's unfinished calculations stay where they are. They'll still be there in the morning. For now, the moss is damp. The organisms have returned to their quiet work. And you are completely safe, resting somewhere within the vast self-regulating machinery of a living world. Good night.